Greetings, my name is Priscila. I'm from the Federal University of Rio de Janeiro in Brazil, and I'm gonna present to you my project entitled Microbial Production of 2,3-butanediol BD and 3-acetylene AC isomers. So, BD and AC are compounds of great industrial interest, and their applications are stereo-specific. Their microbial production usually generates a mixture of isomers, and the microorganisms utilize this pathway shown here. So we have the canonical pathway for PDNAC production, and we can observe that acetylene can be directly generated from acetylactates or through diacetyl, and PD is produced from AC. The two major enzymes involved in isomers production are butanediol dehydrogenase, here called BDH, and diacetyl reductase, here called DAR. So BDHs are very well studied enzymes, and there are three types: R, S, and meso-BDH. RBDH is a particular enzyme, very different from the other two, while S and meso-BDH are very similar to each other, as they are both short-chain dehydrogenases with the same catalytic residues, similar lengths, and also the same stereospecificity, which made us wonder which are the features that can separate them into two different types. Besides that, DAR is not very well studied, which is a problem itself. So, in order to investigate the aspects related to these three enzymes, DAR, S, and meso-BDH, we conduct a comprehensive literature review involving papers and databases, combined with a phylogenetic analysis. So, here showing briefly the literature highlights, we constructed this panel showing all the possibilities. That means all the enzymes and reactions involved with the canonical pathway that can result in the isomers of BD and AC. Here we can see that S and meso-BDHs are able to do the same reactions, which was expected in a certain way, as they have the same stereospecificities. But it reinforces the need to understand their differences. Besides that, we have here DAR in the reaction in the conversion of diacetyl to AC, which was also expected, but as the DAR activity is not very clear, we included these enzymes in the computational analysis. So, we constructed a phylogenetic tree. We constructed this phylogenetic tree with sequences of annotated meso, S, BDHs, and DAR. Not only annotated sequences, but also sequences with experimental evidence. And here we observe the formation of 4 different groups, very well separated. So, meso and S BDHs could clearly separate, but it was important to understand which factor led to this. And we found in the literature, non-catalytic residues triads that could affect the substrate recognition of these enzymes, which would be related to some different stereoselectivity between them. So here, meso-BDH has the triads colored in black and gray, which similar to each other but very different from the S-BDH triads colored in blue. Besides that, we observed that the only DAR with experimental evidence grouped separately, showing that it's actually not a BDH as expected, and it was the first time that the non-catalytic triad of DAR was reported in the literature. So Here in this work, we brought attention to the particularities of these enzymes, and we hope that these information can be helpful for future uh, studies around the improvement of the isomer production.